think we'll get started. A pleasure to welcome you all to this afternoon's webinar. My name is Robert Byrne. I'm a cardiologist working at the Royal College of Surgeons of Ireland in Dublin. And it's a real pleasure to be joined by Georgios, Siontis and Arjola, Bano, who are the speakers for today's webinar. And I'll show you the program in a moment. This is one of a number of core MD webinars which have taken place over the course of 2023 here, and you can see details of the webinars we've had on different topics over the course of the year, and these are also available to watch on playback on the Core MD website. Today's topic is one we hope you'll find of interest. The focus of the webinar is on diabetic and cardiovascular devices. And in this regard, as I mentioned already, it's a great pleasure to be joined by Arjola Bano from the Institute of Social and Preventative Medicine, which is located at the university in Bern, and she also works in the Inselspital in Bern, and Georgios Siontis, also from the Universitätsspital in Bern, and he will be our first speaker this afternoon. Pleasure also to welcome Lorna McGovern, who's joining us as a panelist and may talk briefly on cardiovascular devices as it applies to surgical heart valves later in the webinar. Finally, before we get started, a brief word to mention the next webinar in the series. There's 3 more scheduled, and you can find details as the agendas are finalised on the website. And the next webinar is 27th of November at the same time, 5 p.m. CET, International Standards for Clinical Investigations of High-Risk Medical Device. Some of the Core MD outputs analysed with recommendations for regulators, and hopefully you'll find that webinar of interest. So without further ado, it's my pleasure now to hand over to Georgios Siontis, who's joining, as I said, from Bern in Switzerland, and he's going to give us an update on cardiovascular devices. Georgios?
Great. So thank you, Robert, for the introduction. Thank you for all of you for attending today the meeting. And today the topic on which I will comment on and present some evidence is the availability and quality of clinical evidence for high-risk cardiovascular devices, which have been available for clinical use in the European Union over the last 20 years. And I will start my talk with a question. Is the clinical evidence always published? And we know that almost among 5,000 clinical trials examined by Science back to 2020, less than 45% had their results reported early or on time to clinicaltrials.gov. And the observed violations cover trials in virtually all fields of medicine. In another empirical evaluation among more than 13,000 trials, registered again in clinicaltrials.gov, which completed between 2008 and 2012, only 13% reported summary results at 12 months. And more specific, for cardiovascular devices, a cross-sectional analysis showed that many clinical trials for high-risk cardiovascular devices approved by FDA remain unpublished. Finally, it has been also shown that more than 40% of post-approval studies had not been published 5 years later. Now in Europe, a key objective of the regulatory reforms is to promote higher levels of evidence before high-risk medical devices are approved. In the past, many devices were authorized without being supported by clinical trials. However, nowadays, basic information and clinical evidence is expected to be in the public domain for any approved high-risk medical device. European investigators have played a key role in the development of landmark high-risk cardiovascular devices, with first-in-human studies performed in Europe, followed by device refinements and iterative studies. Examples include the implantation of the first pacemaker, coronary artery stents, transcatheter aortic and mitral valve implantation devices, as well as tricuspid repair. Currently, more than 500,000 types of medical devices are on the European Union market, and these medical technologies are regulated by laws that govern the safety and performance of devices across their entire lifetime. The classification of medical devices in Europe uses a risk-based system taking into account the vulnerability of the human body and the potential risks associated with devices. All devices for use in the heart, central circulatory system, central nervous system, joint replacement, spinal disc replacement, and reasonable implants are classified as high-risk Class 3 devices. For the evaluation of medical devices, guidance documents and recommendations of the design, analysis, and the reporting of trials exist. For example, ISO standards describe the development stages to assess the clinical performance and safety of those medical devices. And further device-specific guidance documents exist, for example, for heart valves and resorbable devices, in which randomized controlled trials as clinical investigations are recommended. Now, the new European Union law governing the regulatory approval of medical devices took effect from May 2021. In this report from the Regulatory Affairs Committee of the ESC, they highlighted the changes took place and the mechanism developed to provide transparency by summarizing the main safety and performance aspects of the devices and the outcomes of the clinical evaluation in a document that should be publicly available. Having this background in mind, and before we dive into the details of the Core MD consortium and the results of our work, I will try very briefly to highlight through two examples of high-risk medical devices, both from cardiology, the complexity and heterogeneity in clinical evaluations of different devices. One of the devices failed after being for considerable time available for clinical use, and the other one, I would say, is referring to a very successful class of devices which changed the landscape of catheter-based interventions. And I will start with a device which was withdrawn from the market after being available for clinical use for considerable time. And this is the bioresorbable scaffolds, coronary scaffolds. In the past, the EAPCI task force reviewed and established a comprehensive list of all drug-eluting stents, coronary stents that had received CE mark back to that day. The group performed a systematic review of the literature of all published randomized clinical trials, evaluating coronary artery stents, and provided recommendations for a new regulatory process for coronary stents. They identified more than 150 randomized trials of coronary stents which had received CE mark that time back. The task force proposed some key steps in clinical development plan for the evaluation of coronary devices with the aim to balance the preservation of patient safety with the avoidance of unnecessary delays in the introduction of innovative technologies for clinical use. A few years later, the EAPCI task force prepared a dedicated report on the class of devices known as bioresorbable scaffolds. The scaffolds were introduced as a novel technology with potential advantages over the metallic coronary stents. It was hypothesized that scaffolds might address late stent failure and potentially eliminate the risk of late adverse stent-related events and maybe contribute to the restoration of physiological vasomotion within the treated segment. In this report, 5 different devices were identified, and the task force provided recommendations concerning the clinical use of the scaffolds and recommendations for preclinical and clinical evaluation before approval of these devices. They also highlighted that only one device, that time back, of the absorbed bioresorbable vascular scaffold, had published data from randomized clinical trials, and this data showed inferior outcomes compared to conventional drug-eluting stents. For this reason, the conclusion was that scaffolds should not be preferred over drug-eluting stents in clinical practice. For the only device with evidence from randomized trials, the absorbed scaffold, we also evaluate how a stepwise approach of monitoring and quantitative synthesis of the evidence over time could potentially identify the time point when firm evidence for safety concerns, even of this relatively rare outcome, becomes available. In that case, 8 randomized trials of more than 8,000 patients were included. And as you can appreciate in this forest plot of cumulative meta-analysis, CMARC was granted in January 2011 without any evidence from randomized clinical trials, but with evidence suggestive of harm related to BVS being already publicly available before FDA approval in July 2016. The rest of the evidence after FDA approval increased considerably the precision of the estimates over time. Finally, the device was withdrawn in September 2017. On the other side, there are successful classes of cardiovascular devices, and one of these is definitely the TAVI. The evolution of valvular heart interventions, either surgical or percutaneous, especially over the last 50 years, is absolutely outstanding. And the technological advantages over a period of only 15 years, I would say it's something unique for TAVI devices. TAVI trials have been a very good example of natural progression from superiority trials to non-inferiority, and it's an example which shows how this transition can take place while it's moving at the same time to broader populations when the trials are appropriately designed, even in the scenario of an innovative alternative treatment. And the accumulation of evidence over time illustrates this safe transition very nicely, moving from high-risk candidates to a broader potential population when at the same time superiority trials are followed by non-inferiority trials. This solid evidence around TAVI over a decade now has been translated into upgrade of the corresponding recommendations in clinical practice guidelines. And at the same time, this rapid evolution in the specific field required sequential updates of what we should consider appropriate clinical endpoints for transcatheter surgical aortic valve clinical trials, indicative of the progress in the specific field. TAVI changed the field of cardiovascular medicine dramatically, and there is no doubt that TAVI fueled transcatheter valve therapies beyond the aortic valve, and actually it served as a blueprint for evidence-based evaluation of devices in this field of cardiovascular medicine. Having all these heterogeneous facts in mind, it is clear that there is a need of better understanding of the evidence used in the past to assess high-risk medical devices before and after CMARC. And this was one of the objectives of CoreMD project, a project which was supported by a grant from the European Union Horizon 2020 call. The CoreMD consortium comprised a unique collaboration with wide geographical distribution across Europe, importantly including national regulatory agents and notified bodies, as well as public health institutes, medical professional associations, academic institutions, and patients. Main tasks were to review existing evidence, to develop new methods for generating clinical evidence, and to investigate how to extract maximal information from real-world data. Our task was to critically assess publicly available clinical investigations used in the evaluation of specific high-risk cardiovascular devices and identify differences in study designs before and after CE-mark approval during a period of 20 years. We had a pre-specified protocol for this study, which was publicly available in Prospero registry and CoreMD website. We followed well-validated approaches to retrieve reports of interest following the PRISMA recommendations, and we used device-specific search algorithms for studies of prospective design, and we summarized information on study design, populations, interventions, and outcomes. The analysis was done on study level, and we followed data-driven approaches to evaluate differences in the distributions of characteristics before and after CE-mark. We predefined 7 groups of class 3 cardiovascular devices of 71 devices put on the European Union market since the year 2000, including bioresorbable coronary scaffolds, left atrium appendix closure devices, transcatheter aortic valve implantation systems, surgical aortic and mitral valves, transcatheter mitral valve repair and replacement systems, leadless pacemakers, and subcutaneous ICDs. By using device-sensitive search algorithms, we identified trials of prospective design, either randomized or non-randomized, which evaluate at least one of the devices of interest. We excluded randomized trials aimed to investigate other medical interventions than the implanted devices and only any other studies of non-prospective design. More than 40,000 studies underwent evaluation with final inclusion of 380 unique studies across all classes of devices. And here you see a summary of the clinical trials characteristics for all 308 clinical trials. Only 19% of the studies were randomized. A protocol registration was available in less than 50%, and the peer-reviewed publicly available protocol was available in only 16%. While more than 80% of the studies report device-related adverse events, event adjudication was only reported in 39%. There was an obvious progressive increase in the number of clinical trials and in the number of included patients over time, with most studies performed in patients recruited after 2015. Overall, almost 100,000 patients were recruited in the 308 trials, including the analysis. Related to the time lag between the time of study publication and CE mark approval, we found that there was not a single randomized clinical trial published prior to CE mark for any of the 71 cardiovascular devices. The vast majority, almost 90% of the non-randomized clinical trials, were published after CE mark approval. We also found that trials with a larger sample size and the longer recruitment periods were more likely to be published after CE mark and more frequent during the last 5 years of our study period. Studies published before CE mark of the corresponding device were published very close to the CE mark date, were smaller in size, and had a short recruitment period, which was not the case for the studies after CE mark. Now, according to the study design, the principal differences between randomized and randomized clinical trials were the larger sample size of randomized trials. The randomized trials were more frequently multi-center, provided power calculations, used the composite primary endpoint, and applied outcome adjudication in the majority of the studies. Surprisingly, sex and age-specific analyses were reported in only 1/3 of the studies. We are also able to assess the risk of bias in the non-randomized studies of comparisons and, of course, in the randomized clinical trials. The majority of those non-randomized studies were at critical or serious risk of bias, but only 16% of the randomized clinical trials were at high risk. In summary, we did not find a prospective clinical trial for 30% of the pre-specified devices. Only 9% of the studies were published before CE mark approval, and only 19% of the studies were randomized controlled trials. Event adjudication was reported in 40%, and not a single randomized controlled trial was available in the public domain prior to CE mark approval for any of the devices of our interest. We concluded that the quantity and quality of publicly available evidence from prospective clinical trials was deemed insufficient for the majority of the CE mark approved devices which were evaluated. The majority of the studies were non-randomized and performed in small populations without provision of power calculations, and none of the reviewed devices had randomized trial results published prior to CE mark certification. It is clear that every new device should undergo systematic non-clinical testing prior to evaluation in clinical studies, which have to be also prospectively designed. Randomized clinical trials are obviously mandatory for new devices, and priority should be given to efforts aimed to evaluate products' net clinical benefit. Also important are the post-marketing studies, which should be also prospectively designed. Now, in the absence of large clinical trials, which is a common scenario, some adverse events may not be known a priori when a new device is used, and in these cases, additional monitoring tools may be needed. We should also keep in mind that over the last decade, we have gained valuable knowledge on different classes of devices, and this knowledge, along with dedicated methods, should be considered to appropriately plan future studies and avoid unnecessary exposure of patients to risks. In this direction, routinely collected data have substantial potential for improving the conduct and reducing the cost of randomized clinical trials, but with some practical limitations like data quality validation issues. Quality, transparency, and reproducibility of research findings are overlapping and supplementary conditions, which are important in all scientific fields, and this is what we tried also to highlight out of this project in the Core MD consortium, specifically for clinical research related to high-risk cardiovascular devices. It is expected that by improving these conditions in clinical research will increase the credibility of the published data and accelerate discovery. This slide is a print screen from the PLOS Medicine website, a leading general medical journal, which I think simply summarizes the key steps for a successful study and what should be considered in every research effort. And this can be summarized in a single term, which is transparency. A detailed report of the systematic evaluation became recently publicly available, and there you can find further details for the different classes of devices and the information we summarized. And to conclude, I would like to thank the whole team contribute to this review and also the colleagues in the core MD consortium for their support over the last more than 3 years now. Thank you.
Georgios, thank you indeed for an excellent overview of this work and particularly the analysis of high-risk cardiovascular devices. We have time planned in for discussion now, and please, I can see an excellent turnout online, and I'm sure you have questions related to Georgios's presentation. So please enter your questions in the Q&A box, and I'll moderate the discussion and put the questions to Georgios. And I can see, thank you, the first questions are coming in already. We'll also have time for some comments from other members on the panel in due course. So let's start with the first question here, Georgios. From Stefan Sauerland, great talk. Could you please say something about clinical studies on those new devices which never reach the market? Is there any duty to publish such negative trials? I understand the MDO rules in the way that transparency is regulated only for CE approved devices. So I think that's a very good question. So it's a great question. It's a really great question. So the first part of the question is whether clinical studies on those new devices which never reached the market. We're not able to evaluate this in our systematic review because we used a reverse approach to identify the studies. First, we defined the devices, and then we went back to identify potential studies available. So I cannot comment on this comment directly. It's unfortunately we did not capture this, and it's not that easy to capture this. On the second part of the question is about whether any duty to publish such negative results. They must be published, these results. They have to publish these results, and this study should be out and publicly available to anyone for to be judged by everyone. And this is exactly the point where the bias is generated. And if these studies are not published, then similar device from the same class cannot be of any, there cannot be for help for any device in the same class. And the answer is clear, yes, this study should be published. Yeah, no, I think that's a clear answer, Georgios. It's clearly an issue. Now, progress has undoubtedly been made if we look back over the last 20 years since pre-registration of trials became mandatory. And while pre-registration doesn't mean that all negative trials will be published, at least it affords a higher degree of transparency. And you're right, the devices that don't have CE mark weren't the subject of the analysis that we did in Core MD. But I think it is very important that these results are published in the public domain. Granted, these experimental devices don't have CE mark, so patients aren't going to be exposed to risk because there won't be access to these devices. But still, of course, they'll guide future iterative developments. On the other hand, devices which have CE mark, where there's randomized control trial data, then it is imperative, if that post-market randomized clinical trial data is negative, that it's made available in a timely manner. There's lots of questions coming in now, and I think we can take some time to address these at this stage. Sally Sennett asks, great presentation. Do you expect things to improve under MDR, and is there a plan to repeat this exercise in the future? That's another great question. We hope that this will be improved, and we need to monitor this. We need to keep an eye on this, and maybe it will be a good exercise to repeat this review in a few years from now. Yeah, I think that's a well-made point. It would be nice to see what the impact of MDR has been, and of course, the requirement for clinical investigation under the MDR is quite different from what was existing previously. And I suppose in the look back that you performed, it was generally pre-MDR devices that you were looking at. Now, another question here. Have you looked at the RCT time versus time that the device was put on the market? What about serious adverse events not seen during pre-market results? Any new during RCT? So, I mean, I suppose time of the RCT versus time to put on the market, you did do an analysis on that, if I'm correct.
Yes, but not for the serious adverse events. This was not captured, actually, in our review.
Correct, but you looked at the time lag between RCT and device going on the market, and one take-home message is obviously the headline result that there wasn't a single RCT available.
Correct.
For CE mark. And then what about the general time lag between CE approval and RCT becoming available? Was there kind of a median value for that? Or?
Yes, I can't recall exactly the number now, but it starts very close after the CE mark approval for most of the devices.
Very good. And I think that data will be available in the full publication as well. Now, another question here from Kareem. Would publications in grey literature be acceptable as well? So I don't know if you want to take that one first.
Mm-hmm. Yeah, very good question. For our systematic review, we decided to exclude this kind of studies. We try to keep focus on peer-reviewed publications where it can be easily accessible from everyone. And that's why we decide not to include any grey literature on this case, or evaluate the publication in grey literature.
Yeah. I mean, it's challenging to find results that were disclosed perhaps in a conference format, but which didn't make it into the published literature and weren't subject to the rigors of peer review. I mean, the short answer is, as you've alluded to in your answer, that there is value in this data. It shouldn't remain in the grey literature, of course. It should be published, but sometimes, for the reasons that you showed in your analysis there, not all of these studies make it into the published literature. And while I'm sure there's value in that grey literature, it is more challenging to abstract this data.
And if something stays on the grey literature, this is not evidence, should not be considered evidence.
Mm-hmm. Yes, yeah, it's for devices where there's a clear gap, I suppose it's the next best information that you have. I mean, in an ideal world, it would never be the case that it just remains in the grey literature, but it's because of the problem that you showed at the start of your presentation, the gap in the literature with negative studies not being published. There is a significant problem there.
Actually, it's the main source of bias in this kind of evaluations and for this kind of studies.
Yes. Lorna, I'm going to maybe come to you for a comment, if you wouldn't mind. Georgios presented the overview of the cardiovascular devices, and in a moment or two, we're going to move on to diabetic devices. But you did a lot of work in particular on surgical heart valves. Are there 2 or 3 take-home messages you could give us from the analysis just of surgical heart valves?
Yeah, well, first of all, thank you for inviting me this evening. So yeah, like you were saying, I did work on one of the 7 classes of cardiovascular devices that Georgios has already referenced. So I looked at surgical aortic and mitral valves. Again, we would have used the same inclusion criteria as the overall project, and looking at devices that received CE mark approval from the year 2000 onwards, and then published data between January 2000 and August 2021. In the initial search for surgical aortic valves, we had over 4,900 articles, and for mitral surgical valves, we had over 3,600 articles. And by the time they were screened and the exclusion criteria was applied, we had 39 aortic and 1 mitral that were included. And then this was across a span of 17 surgical aortic valves, and then we included 4 surgical mitral valves. And very similar to what Georgios was saying, we found overall that the quality of the evidence, the scientific evidence supporting the use of surgical aortic and mitral valves was quite poor, and it was from largely from small industry-funded non-randomized control trials. And in particular, the follow-up, the median follow-up would have only been 24 months, and the majority of studies were classed as having a high or serious risk of bias. And like Georgios was saying, there was very little power calculation or pre-registered study protocol. So I suppose it was much in line with the overall findings from the cardiovascular devices in CoreMD.
Very good. Thanks for that, Lorna. It's, I suppose there's, from your presentation, Georgios, we can see that you showed an example of a technology where the evolution of clinical evidence seemed to work very well, which was TAVI. In some respects, that conflicts a little bit with the data that's available on the surgically implanted heart valve. So we recognize that there tended to be a somewhat different model that was used with many studies using objective performance criteria for surgical heart valves in the past. But I don't know. When you look across the classes of devices, are there lessons from the TAVI experiences that could be more widely adopted? Some of the limitations will be the same in that there weren't any randomized trial data available before CE mark, if I'm not mistaken. But there's also obviously some messages that could be generalized and of importance for those investigating class 3 devices.
Yes, actually, I think one of the key points in the TAVI story since now 12 years now, maybe more than 12 years now, is that the way that they start implanting the TAVI devices until the first randomized control trial came out. They start from high-risk patients that had no other option, or the surgical approach was not a good option for them. The next step was just high to intermediate risk patients, and then they tried to implement this in a randomized trial. And this information was used step by step to build the next trial. And this is the key point with the randomized trials in TAVI in TAVI field, in this class of device of TAVI. They are not compared to stents trials. They're not that many trials. They're very, very limited trials. But I think all the trials have been built on the previous information have been available for this class of devices. And this is the successful story out of this. There was not information that was not used, and this information was used properly to avoid this a lot of efforts without any benefit for the patients.
Yeah, I think that's a well-made point, and certainly if you contrast them with the other example that you use, bioresorbable scaffolds, I mean, in many respects, we had so many papers in bioresorbable scaffolds telling us which endpoints to look at, suggesting even new endpoints for this device. I mean, you showed very well in TAVI the nice evolution of VARC1, VARC2, VARC3, but having clear definitions is only part of the story. And I suppose in bioresorbable scaffolds, you might say they started with the lowest risk patients, in some respects are highly selected patients with straightforward vanilla type lesions or straightforward lesions. And perhaps that's a part of the problem, because the findings then weren't had lesser degree of external validity when you went to use the devices in undifferentiated patients in clinical practice.
Exactly, which was not the case in TAVI devices, because it's exactly what you described. This was not the case in TAVI device. In TAVI devices, they started with let's say the worst case scenario, and they implemented the same technique to less risky cases. And in that case, it was proven successful, which was not the case for BVS.
Yeah. I'm keeping an eye on the clock here and questions coming in. I suppose one thing, I don't know if you have a thought on this, Georgios, but we saw the week before last, or the start of last week, the long-term follow-up from some of the TAVI studies comparing TAVI against surgical intervention. We had long-term from the low-risk partner trial presenting 5 years results and published in the New England Journal of Medicine. And it cuts across, I suppose, the topic of Lorna and your presentation as well. But one thing we do see in these studies, it's very interesting. No matter what study you look at, the degree of follow-up is less in the patients who are randomly allocated to surgical intervention rather than transcatheter intervention. I mean, we're used to seeing this across the cardiovascular literature for many years. We saw it first in the Syntax trials comparing stenting with CABG. And I don't know if you have any insights or explanation into what the reason might be, because certainly the longer term follow-up of these patients is an important part of the story.
Yes, and we also have extensively discussed all this on this point. It's a very good point. As you said, has been observed the same pattern in other surgical versus non-surgical trials, like CDAX is one of them, and not only for heart valve disease, but also for coronaries. And it's always the same pattern. And the question that arises from the latest results from partner trial, actually, it's whether the analysis approach was the right one for these patients. Because a considerable number of patients who had been randomized for surgery, they never received a therapy. These patients were not being included in the analysis. And this raised some concerns. And there are some, I think in the end, soon we'll see some hidden evidence out of this data. And this information will come out.
Okay, so I'm conscious a little of the time. I'd like to thank Jean-Baptiste from EFORT for helping us with the coordination of the webinar this evening. And I think this is a good time to pause the discussion on cardiovascular devices now. And we can pick up if you have questions left in relation to the cardiovascular devices. We can pick up on those after the next presentation, which is from Arjola Bano. I'll be also joining us, as I said, from Bern in Switzerland. And she's going to give us an update on high-risk medical devices for diabetes care. So Arjola, over to you. We're looking forward to your presentation very much.
Hi, thank you very much, Robert, for the introduction. So indeed, I'm going to have an overview in this presentation on our findings of the systematic review and meta-analysis that we performed on high-risk CE-marked medical devices in the field of diabetes care. So the first focus of this presentation will be a background, some background slides, which are an overview on medical devices and high-risk medical devices in the field of diabetes. So during the recent decades, the management of diabetes has changed profoundly, and also the ability to control glucose levels has improved significantly. The development of several medical devices, including here automated insulin delivery systems, insulin pumps, as well as implantable continuous glucose monitoring systems, have greatly contributed to this progress, thus representing a paradigm shift in the field of diabetes care. And Of course, there are multiple devices in the field of diabetes, and some of them can be also considered high-risk medical devices because they either involve automated insulin delivery systems, so automated drug delivery, or implantable components. And of course, there are some regulations based on the medical device regulations in Europe, which include also specific rules of high-risk medical devices, and also specifically in the field of diabetes. And based on these specific rules, we identified first of all, the classes of devices that apply to be considered as high-risk medical devices. And then our aim was also to identify the specific devices within these classes. So after searching the literature and multiple discussions, we came up with three main classes of high-risk medical devices in the field of diabetes, which include the implantable continuous glucose monitoring systems, the implantable insulin pumps, as well as the automated insulin delivery systems, including here the hybrid and the fully closed-loop systems. And the principle of these high-risk medical devices or medical devices in the field of diabetes, especially the automated insulin delivery systems, is based on several components that are crucial in these devices. So the first component is the glucose sensor, which sensors the glucose levels and it is inserted into the skin, and it senses what are the glucose levels subcutaneously in the interstitial fluid. Then this information is communicated to a control algorithm device, which together with additional information based on personal settings of the patient and also additional information regarding the prediction of glucose fluctuations, this control device algorithm is then linked to the insulin pump, which is inserted and which then produces or delivers insulin in order to keep the glucose levels on the blood within a target range. So this is the principle based on which these devices work, and there are multiple of them. So, for example, there is in the first figure, there is the AccuCheck Diaport system, which is an insulin pump with a cannula, and then the others, Medtronic MiniMed or TSLM or the Diaboloop, are also examples of insulin delivery, automated insulin delivery systems. And then based on these classes, so then we came up with a specific list of medical devices, and we identified specific classes of devices, the manufacturers, and also the CE mark approval dates for these devices. So why are we actually conducting this project? At the moment, the clinical evidence that is submitted for a regulatory approval is not transparent. And a systematic review and meta-analysis of the evidence on the efficacy and safety of high-risk devices approved for managing diabetes in Europe is lacking. So this is the reason why in the setting of the CoreMD project, which also integrates other classes of high-risk devices, our aim, a specific aim, was to conduct a systematic review and meta-analysis assessing whether the CE-marked high-risk medical devices for diabetes management are safe and effective. And this process included reviewing of study design, statistical methods, multiple reported outcomes, and also the overall quality of evidence in the field of diabetes. So we started with publishing a protocol paper in PLOS-MED and also Prospero, where we highlighted our main strategy for this project. And this is our team, which also became a bit larger with the time. And then we also performed a search strategy. We decided after multiple consultations with our medical librarian, the databases based on which our search strategy would be more efficient. So we included Medline, Embase, Cochrane, and Web of Science, and also we had also a search at the UDAMED database, and we were also based on information from Swissmedic, notified bodies, and manufacturers. The inclusion criteria, so we decided to include the studies evaluating efficacy and or safety of high-risk CE-marked medical devices for diabetes care. We included observational and experimental design studies, both pre- and post-market, and studies performed in humans only. The exclusion criteria, so we excluded proceedings, systematic reviews, and meta-analysis, which were already performed, conference abstracts, or animal studies, or letters to the editor. So non-original studies basically were excluded. When defining our research strategy, it was also very important to identify all the elements of the research question. So our study would include participants, basically patients with diabetes, both pediatric and adult populations, studies that compared high-risk CE-marked medical devices for diabetes with any other device or class, including here also no intervention, so also pre-post studies were included, and also studies. We chose a wide range of outcomes for efficacy. We chose outcomes related to glucose control, such as glycated hemoglobin and also time in range. We also included acute and chronic glucose-related complications as outcomes, and we included safety outcomes such as severe hypoglycemia, diabetic ketoacidosis, device-related serious adverse events, device deficiencies, and field safety notices. And here is an overview of our search strategy. This is how it looks like only for one database, which is Embase. With the help of a medical information specialist, we defined all the blocks, and then in the end, we converted this search into the other databases. With regard to the methodology, so our study includes two components: the systematic review part, which provides information on the narrative synthesis of the findings, and also we provided descriptive information into summary tables with regard to multiple aspects of the included studies. But also our study includes the meta-analysis component. And we are also planning to perform other meta-analysis because the data is very large. But the main meta-analysis incorporate the automated insulin delivery systems, which also include the largest number of studies in the field. So we performed a meta-analysis of randomized clinical trials that compared these systems, automated insulin delivery systems, with any other therapies for the treatment of diabetes. And then we performed another meta-analysis of studies that compared outcomes, for example, efficacy outcomes, before and after the utilization of these automated insulin delivery systems. We also assess the heterogeneity in our meta-analysis, and we also assess the quality of evidence. Here, with regard to the quality of evidence, the way we assess the quality of evidence was also adapted to the type of study design that we had included. So for randomized clinical trials, we use the Cochrane risk of bias assessment tool. For observational studies, we use the Newcastle-Ottawa scale, whereas for non-randomized interventional studies with no control group, we used also a specific tool from the National Institute of Health, NIH. So after selecting the studies, we started with more than 6,000 records. After excluding database, we came up with more than 4,000 records to be screened. And then we came up with more than 1,000 full-text articles to be screened, after which we identified a total of 117 eligible studies to be included in our systematic review on efficacy and or safety. Here, I also wanted to mention that we also have information on usability outcomes, but because of the feasibility of the project, we will decide to do it as a separate project. So we also collected information on patient-reported outcomes and some other usability outcomes. Then, if we focus on the efficacy and safety, we came up in the end with a total of 98 studies focused on automated insulin delivery systems. A total of 8 studies focused on implantable insulin pumps and 11 studies focused on implantable continuous glucose monitoring systems. So the majority of studies is really focused on relatively recently, so starting from 2009 to 2022, when our search ended, then we noted that 41% of the studies had an industry funding, 18% of the studies were pre-market, and the rest were post-market studies. The majority of the studies were observational, with 46%. We also included RCTs. They were 30% of the total number of studies, and non-RCTs 24% of the total number of studies. So the main results, we can see that the median sample size was relatively small, with 52 participants. We included the eligible studies were predominantly performed in type 1 diabetes patients. They were predominantly performed in adult populations. The follow-up time was relatively short, with a median maximum follow-up time of 13 weeks, and also 47% of the studies had a comparator group. If we focus on efficacy outcomes, we have a number of studies reporting on glycated hemoglobin, time in range. What was interesting is that we didn't identify any studies reporting on chronic glucose-related complications, which also is in line with the short-term follow-up of eligible studies. Then a brief overview on the results of a meta-analysis of RCTs that compared automated insulin delivery systems with other anti-diabetic treatments, for example, standard treatment of diabetes or insulin, basal bolus therapy, and so on. So if we focus on on the studies that looked only at glycated hemoglobin, we see that the pooled mean difference of minus 0.22, showing that basically there is an improvement of 0.22 points of glycated hemoglobin in those RCTs that used automated insulin delivery systems compared to other treatments. And also there is an improvement in time in range, with approximately 9% more in automated insulin delivery systems compared to other anti-diabetic treatments. Then, if we focus only on those meta-analyses of studies that compared outcomes, such as HbA1c and time in range, before and after the utilization of automated insulin delivery systems, we also see a similar trend. So there is also an improvement after the utilization of automated insulin delivery systems in both glycated hemoglobin and time in range. Then, when we focused on safety outcomes, for example, including diabetic ketoacidosis or severe hypoglycemia, or various device malfunctions, or local adverse events, infections, and so on. If we focus only on safety outcomes, a total of 71 studies reported on at least one safety outcome. What was remarkable in this category of studies is that the incomplete reporting. So there is many studies that reported only on one safety outcome, but not on others. And also, if we just focus on the results that we have, taking into account the incompleteness of the data, the frequency of safety outcomes was generally similar between the intervention and control groups. We also, as mentioned before, we performed a risk of bias assessment. And if we focus only on randomized clinical trials that were assessed with the ROB2 assessment tool, we can see that the majority of studies are on the gray zone. So basically, the majority of studies have a moderate risk of bias. and the rest is shared between high and low risk of bias for RCTs, whereas for observational studies, the a little bit more than 50% was high risk of bias, and the rest was low risk of bias. So the conclusions from our results at the moment are that the CE-marked medical devices, and in particular the automated insulin delivery systems, improve glucose control. So they improve glycated hemoglobin as well as the time in range. So time within which the glucose levels are between 3.9 and 10 millimole per liter. However, there are no studies that reported on chronic glucose-related complications, and also safety outcomes were partially reported across the studies. Overall, the evidence is characterized by small studies with short follow-up time and methodological heterogeneity. And also there is a need, of course, for developing the standards for future investigations, thereby improving the study comparability and transparency of the findings. And in the end, I also wanted to thank the whole team that has been dedicating and working on this project, and also the organizers of the CoreMD project, which is very, very interesting. Thank you very much.
So thank you very much, Arjola. And this presentation is now also open for discussion. Delighted, as I said, to see such interest in the webinar, a large turnout this evening. So please, if you have questions, I'm moderating the Q&A box here. So please just enter your question, and we'll put it to the speakers or panelists. Arjola, when you have a look at the presentation in relation to the cardiovascular devices and then the data that you have just shown us, are there any commonalities that you think are visible in both of the data sets?
So there are. The type of search that we performed is similar, but at the same time there are also differences across the way that we did the search. For example, the study designs that we selected, we were a bit broader, I think, in the study designs, including also observational studies and non-randomized interventional studies. So if we would want to compare results or preliminary results, we need to take also these aspects into account. But then when looking at some specific details, for example, information pre-market, post-market, or age included, or some other specificities, I think there were also some similarities into between the cardiovascular devices, orthopedic devices as well, and diabetes devices. I don't know, Georgios, if you want to add something in this regard.
Yes, surprisingly, we have similar observations. Surprisingly, yes, and we identify similar limitations in the current body of evidence. And yes, that's true. It's we have similar findings at some point. Yes.
Certainly it strikes me, listening to both presentations, that there are many commonalities. I think there were some differences in the search strategies, but also plenty of similarities in how the searches were conducted. And if you look at the mix between RCT, non-RCT, probably not that far apart, ranging between 35%, I think, in your data set, and closer to 19-20% in the cardiovascular devices. So certainly a minority in both groups, also a preponderance of small studies. So I think certainly there are commonalities. There's a question here building on that from Robert von Boxtel, who's asking, who's thanking you, Arjola, for your presentation. Is there a need based on this to develop standards? Will there be publications on these standards from the team on the last page of your presentation?
Yeah, that's a very good question as well. I think that's the major challenge, because the work that we did so far is kind of putting everything together, all the evidence, and trying to understand what is out there with all the limitations that can be into it. For example, the risk for example, gray literature may be missing, or negative studies may be not published as well in our case too. But I think the advantage of our study is that we provide a large overview of what is out there, and the real challenge is actually into transferring all this knowledge into providing the right recommendations for the future studies. And of course, we learned from these relatively small studies that are out there. So definitely we need longer follow-up studies. We definitely need more, for example, cost effectiveness data. And also we need a larger overview of balancing all the outcomes together and trying to understand what is the actual evidence. And also what we can learn in terms of study design. What can we benefit, for example, from real world data? That's why we also did an integration of all study designs, and we were open to wanting to know what is out there in terms of observational or non-interventional studies, and also interventional randomized clinical trials. I think this knowledge can also help to understand what combining all the advantages and disadvantages of various study designs together. So basically, in one sentence, the challenge is still there into telling what would be the recommendations for the future, basically.
Yes. Okay. Yeah. Well noted. And I suppose these technologies, some of them have been around for a while. Some of them are newer technologies. But are there any documents or perspectives published in the literature already saying, this is the approximate sample size that should be investigated for these classes of devices? I mean, Georgios showed in relation to coronary stents, which was a relatively mature area of medical device technology, you were able to say, listen, these are the numbers of patients that we'd need to be seeing in various stages of devices. Are there any areas of the three different device groups that you studied where you said these are sufficiently mature that we have a good handle on what numbers of patients should be enrolled in non-interventional or interventional studies?
So I'm not aware of any numbers that can be ideal for the specific designs. However, I also think that these power calculations are always or systematically performed in most of the randomized clinical trials, for example, that they perform a power calculation, and then this sample size would be basically enough to tell whether or not there is an association and so on. However, I think that these numbers in general are also arbitrary and tend to have their own limitations in the way that they are calculated. And yeah, there is also a gap in the literature of diabetes with the knowledge that I have, that probably we also need to have more data on, for example, on directions, which numbers in which specific study designs would be more efficient to then conduct a study more efficiently. Yeah.
Well noted, and no, you're completely correct, particularly the point in that it is not realistic to think that there will be a one-size-fits-all approach, but there'll be certain parameters within which those designing clinical investigations can work. Alan Fraser is making the comment in relation to the last question that the CoreMD consortium will help with recommendations for standards, but ultimately those are the responsibility of regulators and ISO committees, which I think is well noted. The proportion of studies in your analysis that were industry funded and how that compares with Georgios, I mean, I suppose it depends a little bit on the stage of the technology. If it's an early stage, if it's pre-CE mark, then not only is this industry funded, but this is an industry sponsored trial where they have the legal responsibility. So if there are a large number of investigational devices, then of course you will have a high proportion of industry funded or indeed industry sponsored studies. What was the proportion in your field?
I think it was around 45%. I can double check the number, but it was considerable, a relatively high proportion of industry funding. And also I want to note that for the studies that we consider as non-industry funded, it's that they didn't report anything. So there is still also in those the rest 50% that there may have been also some industry funding that is not reported actually in the study, or that we did not manage to find this information in the paper itself. So and I fully agree that the diabetes area, also as compared to the cardiology field, is with this specific high-risk medical devices is relatively new, and that can also explain this proportion of high industry-funded studies.
Well noted. It's a parameter I think worth examining. It's difficult to interpret at a high level. Studies that have industry funding available often have more funding to allow incorporation of the classical metrics in a study that are associated with reduced risk of blinding, maybe more sophisticated ascertainment follow-up protocols or adjudication protocols, maybe more facility to introduce blinding in terms of outcome assessment. But of course, then there are other risks of bias that exist, and I think that's why it's important as a community that we have a balance between studies which have industry funding and those which are truly investigator initiated, but also have high quality metrics.
What would be interesting in our specific analysis for the meta-analysis is to do some sensitivity analysis, which I think would take their time. So for the studies that we have included in the meta-analysis, I noticed that there was the heterogeneity in our results, in our pooled estimates, as expected, was high. So now the next step is to explore potential sources of this heterogeneity, and one of the sources can also be related to the industry funded versus not, with the information that we have, which may be also not 100% correct, but with what the studies themselves report, we could do this stratification by industry funding or not, and then see whether the results can have a change or not. And also this publication bias analysis could also be part of the additional analysis for our meta-analysis, to see if the negative studies, for example, are not there or are published less as compared to the studies providing positive findings, which can also be the case.
Very good. Perhaps time for just one more question before we close. Keeping an eye on the clock, Stefan Sauerland again, did you compare studies done in children versus those done in adults? Is your CT design any less common in the pediatric setting?
Yeah, that's actually the case, but we also, together with what I was mentioning before, this is sensitivity analysis based on industry funding. We also plan to do sensitivity analysis based on children versus adults, and we have all this information. And indeed, the children randomized clinical trials are the most challenging to be done. But now there are some new devices coming out that are focused or that can be also used, such as fully closed loop systems that can be also used in children. or starting from the age of 1 year. So it's also absolutely useful to have a look on those devices as well and reanalyze the data that we have.
Very good. Well, I think that's a good point on which to close the discussion. We're right on time, and I'd like to thank you and also Georgios and Lorna for your presentations and comments, and also to Jean-Baptiste and Aoife for the support of this webinar of the CoreMD Consortium. As we mentioned at the outset, this is being recorded and will be available on playback on the CoreMD website over the course of the coming days. If you're not familiar with the CoreMD website, please do check it out. A number of reports from the consortium are available, and you can also see the number of the webinars which were already conducted earlier on this year to review. So I think with that in mind, all that remains for me to do is to remind you that the next webinar will be Monday the 27th of November, same time, same channel, I suppose. International Standards for Clinical Investigations of High-Risk Medical Device, the CoreMD analysis with recommendations to regulators, and you'll find further details of that presentation online. And thanks to all of you for joining this evening and for your interactivity and questions, which we much appreciate. So thank you and have a nice evening.